Alright, ladies, happy fall break. Although notes was not my primary plan for this online school day, we've gotten a little bit behind of where I'd hoped to be right now for various reasons. So we're going to use today to kind of catch up. So I have some notes here that'll kind of wrap up what I had hoped to do previously and kind of move us into the next section as well. So to start out with, I know that we already covered this slide, and so I figured I'd start with a little bit of a recap, which is that, you know, remember we're talking about the exchange of materials, getting things into or out of the cell through the membrane. And so this slide, I just wanted to remind you that some molecules, particularly small ones that are nonpolar, and some of these small polar ones as well, they can move directly through the membrane, right through the phospholipid layer. Other molecules that are charged or larger can't go directly through the membrane. So if you remember, we talked about these ones that can move through the membrane last class. So when we look at these molecules that can go directly through the membrane, remember that what they do is just basic diffusion. Right, going from high concentration to low concentration across the membrane. And you have to excuse this background noise. Samson is going absolutely nuts. I'm going to take his collar off, I think. And so these molecules will diffuse, and they will diffuse until from high concentration to low concentration until we get to a roughly equal value. And we will say that we are at dynamic equilibrium. But remember, at dynamic equilibrium, we don't mean that the molecules will stop moving. We just mean that now they're going to move back and forth at a roughly equal rate to maintain a roughly equal concentration on either side of the membrane. Now, hopefully you also recall this slide, which took a look at osmosis, or the diffusion of water. And so remember that water molecules also will move from high concentration of water to low concentration of water. But we oftentimes talk about not the concentration of water, but the concentration of solutes, or stuff dissolved in the water. So water is going to actually move to where there is more solute. So as we talked about last time, remember that water chases solutes. So if we start here in the middle, isotonic solution equal solutes inside the cell and outside the cell. So you'll notice that water moves equally both directions, and the cell maintains its shape. Hypotonic solutions are going to have less solute outside than inside. Right? And so since water chases solutes, water will go into the cell, and the cell will swell up and potentially burst. Hypertonic solutions have more solute outside the cell than inside. So water is going to rush out of the cell, right? Water rushes out, cell swells up and shrivels up and is also non-functional. So you got three different tonicities to look at, but just remember that water is going to move to where there is more solute, or more stuff for it to dissolve. Alright, and here we are at this slide again. Now, the reason I brought it back is because now we're going to shift our focus to these molecules right here. Because if the molecule cannot freely cross the membrane, then we need transporters to help it get across. So in other words, we're going to need a protein to serve as a doorway here for things to move through. As we talked about in class, a ghost, maybe like these guys, can move directly through the membrane. But humans can't move through walls, right? Or ghosts can move through walls, like these things can move through membranes. Humans can't walk through walls. So for us to do this, we're going to need some sort of window or door to pass through. And these larger molecules are going to need a transporter for that. Now, these transporters can come in a couple different types. They either can allow diffusion to go the direction we would expect it to, from high concentration to low concentration. Or they can help move or push things the opposite direction, right, against the concentration gradient, from low to high concentration, backwards. And if they do that, they're going to require some energy. Okay, so the first group there that you saw in that last picture, a passive transporter or passive transport allows for diffusion of substances down their concentration gradient, as we would always expect it to do with diffusion. And because this is the natural flow of the molecules, this does not require any extra energy or anything like that. The difference here, the catch here, is just that we need a transporter now, right? We need some sort of channel to go through the membrane, because these molecules can't just move through the membrane on their own. Now, sometimes we also refer to this as facilitated diffusion. So if it is facilitated, it has a helper, right? It is something to facilitate it. It's going to be some sort of helper. Now, the other option that you saw in the little orange box, or no, I think it was a blue box on the last slide, is active transport. Diffusion of substances against their concentration gradient. So instead of going from high concentration to low concentration, these things are going to go from low concentration to high. We're going to have to push these, or pump molecules, if they're going to go the wrong direction. And if you're going to pump molecules, you are going to need extra energy. So an active transporter is like if we had 100 people in our classroom and I wanted more people in there, so I started to actually physically shove people into the classroom. That would be like active transport. Sometimes the cell needs active transport to maintain its homeostasis the way it wants it. If you remember too from the first day, we talked a little bit about how equilibrium doesn't always mean, or homeostasis doesn't always mean, equal balance. Right? The cell might actually want more of something outside the cell and less of something inside. And if that's the case, it needs to use active transport to push that stuff out and maintain that higher concentration on one side versus the other. In other words, active transport will sort of resist the natural flow that diffusion would give us. Now this graph is really important, and what I wanted to point out to you with this graph is that if we need transporters at all, so whether we're talking about active or passive, if we need a transporter, notice what can happen, right? So we'll start out here with non-mediated transport. That's basic diffusion, like we saw the other day in class. As long as you continue to increase the concentration of materials, you will see diffusion continue, right, exponentially. But if we need a transport protein, in other words, if we need to mediate transportation, or if we need to facilitate transportation, then those transporters can fill up. Ooh, that's bad. They can fill up. And if that happens, we end up with a curve like you see here in red, kind of like when we learned about enzymes. We said the enzymes can all fill up so they reach a maximum. The same is true for transport. We can reach a point where all the transporters are filled, and then we can't go any faster. So if you're using a facilitator, a transport protein, then the rate of diffusion, or the amount of diffusion, can level off. Now I'd like you to know one example of active transport, and that is a particular pump called the sodium-potassium pump. Now the sodium-potassium pump is sort of a dual action pump, moves ions one way and the other way. And it does this to maintain membrane potential. Now what that means is a difference in charge across the membrane. Most cells, really all cells, are going to be a little bit more negative inside. So if this is a cell right here, they're going to be a little bit more negative on the inside and a little bit more positive on the outside. And the cell has several different functions, several different mechanisms to make this happen, but one of the major ones is the sodium-potassium pump. And this difference in membrane potential is most important to nerve cells. So I'll put especially nerves require this difference in membrane potential, but pretty much every cell in your body will have this. Now the way this pump works, and we've got pictures on the next slide, is that first it will push 3 sodium ions out of the cell, which is against the sodium gradient. And then after it's done that, it will bring 2 potassium ions into the cell, which is also against its gradient. But also notice what I'm doing here. I'm sending 3 positives out, but only bringing 2 positives back in. So slowly, I will make the exterior more positive and the interior a little bit more negative. Okay, so here's what this looks like. If we start up here at step 1, notice that 3 sodium ions are going to move into this pump and get pumped from the intracellular area to the extracellular area. And again, this is against the gradient for sodium. So sodium concentration is actually higher out here to start with, and we're going to add more sodium to that. Now, in order to do this, it's active transport, so we're going to require some energy in the form of ATP, and we're going to use that energy to push the sodium out of the cell. Now, this causes a shift in the shape of the transporter, so now it can welcome in 2 potassium ions. And again, potassium is actually higher inside the cell, so we're going to be pushing more potassium against its gradient as well. So the potassium moves in, shape change occurs again, and the potassium is released inside the cell, and now we're all set up to start over again. And we can pump 3 sodiums out and 2 potassiums back in. Now again, the point of this is to maintain on the membrane a little bit of a difference in charge, and to maintain what the cell wants, which is a higher concentration of sodium outside the cell and a higher concentration of potassium inside the cell. Now, I thought about doing more of an in-depth discussion of the disease cystic fibrosis to kind of go along with our clinical medicine stuff, but instead I just opted for a quicker discussion of it. But I want to bring up this disease, which many of you I'm sure have heard of before, because it is an interesting example of how important these cell transporters can be. Now, cystic fibrosis is very common genetically. It is a genetic disease, so you're born with it, and at this point there's no cure for it. It affects a lot of Caucasians, particularly of European descent. So like it says here, 1 in every 3,200 Caucasian births is born with cystic fibrosis, so that's actually fairly common in terms of a disease. Now, someone with cystic fibrosis has chronic cough and breathing issues. They get a lot of lung infections, and they have difficulties with their digestion, and end up with kind of malnourishment because when they eat foods, they don't absorb all the nutrients. Now, this is the result of a defective transporter. So one single type of transporter that's found in 2 places in the body is defective or does not work in someone who has cystic fibrosis. And because of that one transporter not working, these people suffer from a disorder that definitely shortens the lifespan and could end up, causing their death. Now, what's going on here is there's a transporter called CFTR. You do not have to know what it stands for, but it stands for Cystic Fibrosis Transmembrane Regulator. Now, CFTR is a chloride transporter that is found in the airways, so in the lungs, and also in the digestive tract. Now, here's what this usually does. Let's say this is your lung surface. So here's like the inside of your body. This is overly simplified, and this is out in the airspace. So we're like inside your lung here, but out where the air is. Now these transporters are built into this membrane. And what they do is they move chloride out of the cells. Now, if you remember from last time, water chases solutes. So when we do this, water follows the chloride out. Now, why is this important, is the question. The reason this is important is because your airways and your digestive tract always have a layer of mucus on them. Now, we're very familiar with this when we get sick, and you're like hawking up a lung kind of thing. But you always have mucus there, and it needs to be there. It keeps that surface moist. It keeps that surface protected. But notice what's going on here. That mucus is kept very thin and watery because chloride moves out of the tissue and water follows it. And when water follows it, we keep that mucus nice and thin. Now, if you have cystic fibrosis, then these chloride transporters don't work. So we don't move chloride out. And if we don't move chloride out, then we also don't move water out. And what we end up with is a really thick and nasty layer of mucus. Now, if you have this really thick and nasty layer of mucus, you are going to struggle to move air through there, and you're going to struggle to move nutrients through there. So, these individuals have trouble breathing, and they have trouble absorbing their nutrients when they eat. In both instances, this could potentially lead to death, right? But what's kind of interesting too is that generally people who die from cystic fibrosis don't actually die from cystic fibrosis itself. They die from infections, and the main infection that presents a major problem for them is pneumonia. The more mucus you have in your airway, the easier it is for bacteria to build up and cause a disease, and the harder it is for your body to fight it off. And so the majority of people who do die, kind of quote unquote, from cystic fibrosis, actually die from pneumonia instead. Now, cystic fibrosis does have treatments. It's better than it used to be. Cystic fibrosis was pretty much a disease that you did not live beyond age 20. So mostly teenagers or younger. Today we see people with cystic fibrosis live into their 40s, but still that's not quite long enough. So we hope that they can continue to find better treatment options for this disease. Okay, so the last thing about exchange of materials is that there's also something called bulk transport. Now this is also active transport, so it does require energy. But instead of just moving one molecule at a time, bulk transport is when we want to move really big things or we want to move lots of molecules at a time, hence bulk. So this could be endocytosis, where we take in a pouch of materials, or it could be exocytosis, where we release a pouch of materials. I'm going to start down here at the bottom. Exocytosis is exactly what it is. There's no subtypes here or anything like that. If the cell basically joins a pouch, a vesicle, with the membrane and releases its contents, that's exocytosis. And this is how a lot of your glands secrete a hormone and things. This is also how a lot of your immune cells work, by secreting different chemicals that help fight disease. Now, when we talk about endocytosis, bringing things in, there's a couple different specific types that we see. This is just the reverse of exocytosis, so you form a pouch and bring something into the cell. If you are bringing in something very large, like a whole cell or parts of cells, then you do phagocytosis, and that's what this picture is of. This is an immune system cell, this big purple thing, actually engulfing these little yellow bacteria. So if you intake a whole cell or a piece of a cell, a large structure, we call it phagocytosis. If you take in something small, or you engulf a little pouch of fluid, we call this pinocytosis. So pinocytosis is pictured down here, where this amoeba is kind of forming a little pouch right here that I'm circling, and bringing just some of that extracellular fluid into the cell. And that's going to bring a lot of nutrients and things with it as well. Okay, now this takes care of exchange of materials. And the Edmodo quiz that you guys are going to take as the second part of your online schoolwork for the day will cover exchange of materials. So nothing else moving forward here about the cell cycle will be on that little quiz. But that quiz will have 10 multiple choice questions that come from the notes up to this point right here.